Hello kids, today we are going to start the revision of chapter cell. Here are few questions given in the worksheet which we will be doing today. So our first question is describe the microscopic structure of the cell. So if we are talking about the microscopic structure, so the cork cell were the first cell to be observed. We can say they were composed of box-like compartments forming a honeycomb structure. The cell organelles were found embedded in the cytoplasm, and these were smaller in size and bounded by cytoplasm. So, this was the answer to the first question. Going towards the next question, what is hypotonic solution? If we are talking about hypotonic solution, a solution which have solute concentration lower than that of cell sap is known as hypotonic solution. Fine. Our next question is question number 3, that is what is isotonic solution. So iso means here same. So we can say a solution having solute concentration same as that of cell sap is called isotonic solution. Got it? Next question, what is active transport? So here the active transport in which energy is required. So we can say the movement of molecules across a membrane in cell against a concentration gradient with the help of ATP. ATP you can say is another name of energy. ATP full form is adenosine triphosphate. ATP units. So it is active transport because ATP are involved here. Next question, why is Golgi apparatus why is Golgi apparatus called the secretory organelle of the cell? Why it is called so? This is because it packages material synthesized in endoplasmic reticulum and it dispatches it to intercellular you can say plasma membrane and lysosomes, and extracellular you can say cell surface. ठीक है, at the target sites you can. So moving towards our next question, what will happen if chloroplast is taken out of cell and illuminated? So, as we know, chloroplast is a semi-autonomous organelle of the plant cells, and on illumination, it can carry out photosynthesis and release oxygen even outside the cell if kept in isotonic medium and provide with raw material. Got it? Okay. Now, moving towards our next question, that is question number 7. Here in the question number 7, it is asked what are the functional region of a cell. So basically we are talking about the functional region. There are three major functional region of the cell. First is cell membrane or you can say plasma membrane. Next is nucleus which is also known as brain of the cell, and last but not the least is cytoplasm which carry different organelles. Got it? So here these are the three major functional region of the cell. Next, moving towards question number 8. What is cell sap? Give its composition. So here liquid content in a vacuole of plant cell is called cell sap. Liquid content in the vacuoles of plant cell is called cell sap. The cell sap contains sugar, amino acid, proteins, minerals and metabolic waste. Got it? Okay. Next question is question number 9. Here the question is what would be no plant. sorry, there would be no plant life if chloroplast did not exist. Justify. So here the question is, there would be no plant life if chloroplast did not exist. Justify. So we can say chloroplast contains pigment. Here we know chloroplast contains pigment, that is chlorophyll, which is responsible for food preparation in plants by process of photosynthesis. Hence, if there were no chloroplast, then there would not be seen any process of photosynthesis, and hence we can say there would not be any plant life. Is it clear to you? Okay. Next, why does spinach look green, papaya yellow, and edible part of watermelon is red? Yeah. Why does spinach look green, papaya yellow, and edible part of watermelon red? So here we can say it is all due to different pigments present in them. Since spinach contain chlorophyll, that's why it is green in color. In papaya, carotene is present, which gives its yellowish to orange color. And in watermelon, xanthophyll is present, which give its color red. Got it? Okay. Moving towards question number 11. Why are peroxisomes mostly found in kidney and liver cell? Actually, peroxisomes contain various oxidative enzymes which detoxify the toxic material. Since the blood carry various toxic substances to kidney and liver, a large number of peroxisomes are present in them to oxidize the toxic material. So here, that's why peroxisomes are mostly found in kidney and liver cells. Next is question number 12. What do you mean by plasmodesmata? Actually, plasmodesmata here we can say are present in the plant cell. And as we know, due to presence of cell wall, the exchange of material between the plant cell is not possible. So therefore, protoplast of the plant cells are connected by cytoplasmic channel through their wall, which are called plasmodesmata. And these channels are used for exchange of material between two cells. Got it? Next question is, what is endocytosis? So here endocytosis is the process, you can say, in which ingestion or engulfment of food and other material is done by folding of plasma membrane, as it is seen in amoeba. We have seen in amoeba, it engulfs the whole food. It engulfs its whole food by folding the plasma membrane. So this process is known as endocytosis. Now our next question is question number 14. Why are Golgi bodies found in large number in the cell which separate digestive enzymes? So here the main function of Golgi bodies is to release protein or enzyme by vesicles. No other organelle has this property, therefore these are largely present in secreting cells. Okay. Next question number 15. What is significance of pores present on the nuclear membrane? So these pores present on the nuclear membrane allow transport of water soluble molecules across the nuclear envelope. RNA and ribosomes move out of nucleus, whereas carbohydrates, lipids and protein moves into the nucleus. So these helps in transportation. Next question. What are esters and spindle fibers? Esters are two star-like structure radiating fiber from centrosome. They help in locating spindle and trigger cleavage of cytoplasm. Spindle fibers are formed from microtubules during cell division. They pull the chromosomes apart and bring them at the poles. These are broader in the middle and narrower at the poles. Got it? So this is the difference. Next question number 17. Do you agree a cell is building unit of an organism? If yes, explain why. An organism is made up of various organ system like digestive system, nervous system, respiratory system, etc. And these organ system are made up of various organs, and here these organs are made up of various tissues, and tissues are group of cells performing the same function. So hence we can say a cell is building unit of organism, because cell combines to form tissue, tissue combines to form organ, organ combines to form organ system, and at last with the combination of different organ system an organism is formed. Next question. If you are provided with some vegetable to cook, you generally add salt into the vegetable. After adding salt, vegetable release water. Why? This is the question. So here we can say when salt is added, a hypotonic medium is created. That is, concentration of salt molecule is more outside the vegetable than inside. Hence, due to osmosis, water from the vegetables comes out. Clear? Next, question number 19. How are chromatin, chromatid, and chromosome related to each other? So, if we are talking about chromatin, chromatin is a thin thread like structure which is composed of DNA. DNA means to say deoxyribonucleic acid, and protein to form a rod like chromatid. So, chromatin forms here from chromatin. Chromatids are formed. Two similar chromatids attached to centromere to form a chromosomes, and these you can say attached to centromere to form chromosomes. Fine. Now here last question of the day is, how is bacteria cell different from onion peel? So here if we are talking about bacteria cell and onion peel, so bacteria cell size is very small, it is about 1 to 10 mm, and here onion peel size is you can say larger than 5 to 100 mm. Next, nucleus is absent in bacteria, and here in onion peel it is present. Bacteria cells are prokaryotic, that they do not have well defined nucleus, or you can say they do not have nuclear membrane, but onion peel is eukaryotic cell. Cell division here take place by binary fission or budding in bacteria cells, but here in onion peel cell division take place by mitosis. These are the few differences which you can write. Okay, that's all for the day. Thank you.